We are now going to talk about what I think, as an immunologist, is the single most important thing that this field has done for human life, and that is vaccination. Um, there are so many diseases that are now vaccine eligible that in previous times were death sentences or drastically altered the course of a person's life. And thanks to vaccination, many patients that would be affected by these horrible illnesses are not. And each year, just from like a fiscal standpoint, vaccination saves us millions of dollars in healthcare costs. So it's really just, I mean, I guess I can't say enough about how much I love vaccination, and hopefully you do too. So let's just talk about what the goals are about vaccination in general. So when we vaccinate somebody, we are hoping to increase their immunity to a given organism. So let's just remind ourselves about immunity in general. Immunity is the ability of an organism, us in this case, to resist an infection or the effects of a toxin by the action of a specific and targeted response. All right. And that response is normally in the form of antibodies. There is one vaccine that actually involves the work of T cells, and I'll talk about that. And certainly, anytime we're talking about antibodies, we're at least talking about CD4 positive T cell help at a stage of it. But really, what we want is a memory response that is mediated by memory B cells that are going to be very long lived in the lymph node, and that when we are re-exposed to that pathogen, can differentiate into plasma cells, which will be able to secrete antibody, because it's really the titer of antibody in the serum that is protective to us. All right, so when we think about immunity, because we're thinking about antibodies, we can kind of think about it in two different ways. We can talk about immunity to a single organism, or we can talk about immunity to a closely related organism. Immunity to a single organism, a good example of this would be like the flu shot for now. Hopefully they're getting closer to like a pan flu shot, but for the moment, we receive a new flu shot every year. Each year, there are different strains of the influenza virus. And the majority of the influenza cases that occur in a given year are going to contain components of a given influenza A or B strain. If you make antibodies to a particular strain, let's say it's that H1N1 strain, the antibodies to that one are not going to protect you from H3N2. Okay, there's no cross reactivity. So that's why you need to get vaccinated each year for the new strains of influenza that could potentially be causing flu that year. Immunity to a closely related organism really relies on cross-reactivity. So there are vaccines that are made of organisms that are similar to another disease-causing organism. A great example of this is smallpox, actually. Thankfully, smallpox has been eradicated. Smallpox is caused by the variola virus, which we have not spoken about yet. Because it's eradicated, but it is still kind of an important organism to think of, particularly from a bioterrorism standpoint. So the variola virus vaccine that protects us against smallpox is actually made from the vaccinia virus. Vaccinia actually causes cowpox, but they're so similar, and cowpox doesn't really make us sick. So they're so similar that we can use the similarity between vaccinia and variola, and the antibodies that are specific for vaccinia will protect us against variola. So this, like I said, this is all based on cross-reactivity. Because the antibodies to variola protect us to vaccinia and vice versa, we can rely on that similarity. In both cases, the response is mediated by antibodies. This is all about antibodies. This is really where they get to shine. Because there's cross reaction with vaccinia and variola, then we have immunity to a closely related organism. There is no cross-reaction between these two very different strains of influenza virus, so that's immunity to a single organism. All right, so in this video, I'm largely going to discuss vaccination, but I don't want to forget about the other way that we can develop immunity. There's two ways you can develop immunity. You can get sick with it, or you can be vaccinated against it. That's pretty much it. Natural infection, by natural infection, I mean you literally got infected. There's a lot of debate surrounding natural infection, particularly from people who maybe are uneasy with vaccination strategies. Some of the arguments are that it may provide a longer lasting infection or a stronger protection. And the thought behind that is that ideally, the first time you encounter a pathogen, you would receive kind of the wild type strain, or the strain that is actively circulating in the population. And then recovery from that strain would confer lifelong immunity to the disease in a healthy individual. And that's because the memory pool of cells created is typically larger following a natural infection. If you are then re-exposed to the antigen, this induces the memory cells to begin replicating and quickly establish a new memory T cell response, as well as effector response, and increase the antibody titer. So that's kind of the thought behind it, is that natural infection leads to a strong natural immune response, which eventually leads to long-lasting memory, right? That is true in a way, but it is not the only way. And when I talk about types of vaccines, there are some live attenuated vaccines that actually kind of follow this same curve. So for example, the varicella vaccine or the measles, mumps, and rubella vaccines, those are live attenuated vaccines. And because they are live vaccines, you still kind of get the same effect as if the patient had been infected, quote unquote, naturally. I think what people often kind of forget is that for some illnesses, a natural infection, while it might be beneficial should you survive it in the long run, the survivability or the post-infectious sequelae are so damaging or difficult that it's actually better to get some immunity on board than no immunity. And that's kind of the premise behind a lot of vaccinations. So vaccination is the other way you can develop immunity. So vaccination literally means that it's the process of generating protective adaptive immune responses against microbes by exposure to non-pathogenic forms or components of the microbe. The key to a successful vaccine is to trigger the adaptive immune response, because remember, we need adaptive immunity. Only adaptive immunity has the capacity to create a memory response. And those memory cells are then able to quickly act when encountering the pathogen a second time and develop antibodies. With vaccinations, there's a couple of ways a person can be exposed. So you can get, and this is the most common, an intramuscular shot, just like your flu shot. There are some, or actually just one, oral vaccine. So this is actually for one of the gastroenteritis viruses, where we actually just swallow it, and that's great because it leads to mucosal immunity in the site where that virus is most likely to be a problem. You could have an ID injection, or you could have a sub-Q injection. ID being intradermal and sub-Q being subcutaneous. These aren't as common, but really IM is the main source of vaccination, followed by oral vaccination. So how do we determine if a vaccination strategy has actually been successful? There's kind of a couple ways we can do this. First off, remember, the main thing we're trying to create are antibodies. Okay, so the first thing you want to look at is the quality of the antibody response. Quality and quantity. So the quantity is actually really easy, and we've talked about quantity where antibodies are concerned a lot. Quantity basically just means we want to be aware of what the titer is in the patient. And even if a patient has been exposed to a vaccine-eligible illness before, you're still going to want to have a good idea of what their titer is if you consider them at higher risk. Quality actually has to do with their avidity. So basically, how strongly are the antibodies that are generated to this vaccine able to bind the real pathogen, the real McCoy? So this is the strength of the interaction of antibody binding to the specific epitope at the surface of the antigen. You can think of avidity as the sum of the epitope-specific affinities for a given antigen. So I vaccinate you, you make a whole bunch of antibody responses. I want to know what the sum affinity of all those antibody responses are, and if they are sufficient to protect you from this pathogen should you see it. So the second thing, quantity, is actually really measuring persistence, okay? So you're vaccinated, let's say, when you're 2. How long after your vaccination are you going to maintain a titer that is protective? Long-term immunity is basically the maintenance of that antigen-specific immune effector and or the induction of immune memory cells that are efficiently and quickly reactivated to immune effectors following pathogen exposure. So if that titer goes too far down and the memory B cells are gone, then that's not a very effective vaccination strategy if it all dies out within, say, a month. But if it takes, 10, 15 years for that to die out, well, then you had 10 to 15 years of protection. All right, so let's talk a little bit about the vaccine immune effectors. So I've already mentioned antibodies a bunch, and that's pretty much what I'm showing here. The antibodies are going to be produced by the plasma cells. There's going to be some CD4 positive helper T cell response because we will see IgG and IgA in particular in vaccine responses, sometimes IgE. So these are made by plasma cells, right? The antibodies are capable of binding specifically to the toxin or the microbe. I like to think of this as a little virus, okay? And basically neutralize or block the toxin or viral attachment to the protein, right? So that's literally what this is showing, that if you've already been vaccinated, and let's say this is the flu virus, then the antibodies are basically binding the viral attachment proteins before the viral attachment proteins can bind to our host cells, and therefore it's not going to be able to get in, and it'll be swept away by our innate immune processes. The other thing that antibodies do is that they aid in phagocytosis. Remember the concept of opsonization? Antibodies, particularly IgG, are really good opsonins, and they help the phagocyte kind of grab onto it, and then it makes it for easy eating. And then the last thing, this is actually specific for toxins, but it actually binds the toxin so that it's not able to bind to its cell receptor, and therefore you're not having that immune response. So that's why I say antibodies are kind of the leaders of the memory immune response. Now, that isn't to say that CD4 positive T cells and CD8 positive T cells don't play a role. They do, it's just not as common. So CD8 positive T cells can actually act to reduce, control, and clear intracellular pathogens by basically directly killing the cells or indirectly through antimicrobial cytokine release. So basically, if you have a cytotoxic T cell and the patient already has the infection, then it will help clear that while the antibody response is underway or getting moving. They're probably the least helpful in a vaccine strategy. CD4 positive helper cells, these guys are helpful. The CD4 positive T cells are basically going to promote the cytokine environment that we need to create specific antibodies, right? So one, they're going to produce a ton of cytokines. If we just think about our Th1 cytokines, we can think about things like interferon gamma and IL-6 and IL-1 and IL-12. And all of these are going to serve to kind of promote the inflammatory response. With vaccination, the stronger the inflammatory response, the better your memory response will be. So CD4s are helpful there just in that they're creating a bigger splash with the vaccine. The other thing that they would do is obviously instruct B cells to become plasma cells, which then secrete the all important antibodies. And they actually tell you kind of which class of antibody you're going to want. So the support of the B cell differentiation is also a key factor in T cells. As far as T cell and antibody responses go, there's really only one vaccine that relies on T cells, and it's the BCG vaccine. This one is actually one for tuberculosis. We do not use it in the United States, basically because it interferes with our public health program for identifying patients who actually have TB, because patients who have received a BCG vaccine will actually get a positive TST result. And so therefore they get identified as potentially having tuberculosis, and we wind up having to go a lot further to figure out if it's an active infection or a latent infection or just the result of vaccination. But it is used in Europe quite frequently. Okay, there are two different strategies by which patients can be immunized: passive and active. I'm going to start with passive because we've already talked about it a lot. Passive immunity is basically when I take the antibodies that were made in this person who recovered from something and place them in this person who is currently sick with the illness. Okay, so I've talked about it before in the context of Ebola with Kent Brantley, who provided basically convalescent serum. But that's what this is. Passive immunity is essentially convalescent serum. Or at least that's how we most often think of it. It's a short-lived transient immunity provided through administration of protective, usually polyclonal antibodies specific for a single microbe to a person that has not had prior exposure to that microbe. These antibodies are produced by one human or an animal and transferred to another. Now, if it's a different species, that brings up a good point. If it's a different species, rabbits, horses, any other species but us, it is a one-time only get out of jail free card. What do I mean by that? I mean that you can only get an antibody administration from this other species one time. Because think about it. Rabbit immunoglobulin is still a foreign peptide. Your body is going to respond to it, and it's going to make antibodies to it. So one time, fine, that's a priming event. But from that point on, you probably have antibodies to rabbit IgG. So if you received it again, you would develop something called serum sickness, which is a form of hypersensitivity that can be very dangerous for patients. Protection using passive immunity basically ends when antibodies break down during a period of weeks to months. There's actually one really common form of passive immunization that all of us have already experienced, and that is maternal antibody transfer. Basically, when you are in utero, when you are being developed as a human, the IgG antibodies that your mother already has in her serum cross the placenta and basically protect the baby. So after the baby is born, for several months, the baby has a full complement of mom's antibodies. Additionally, moms who breastfeed pass on IgA to their infants, and that can be another form of passive antibody transfer from mother to child. So basically this one will protect against like gastrointestinal infections, and this kind of protects against whatever mom's got IgG antibody to. Over time, about 3 to 6 months, the IgG antibodies will wane, and the infant actually has a period of like hypogammaglobulinemia where they're actually a little bit short because they really haven't started making their own, and moms have started to die off. Okay, so now let's just talk one more thing about passive, and that's when do you use it? There's really only a couple places you know you're going to use it. First, you can use it to prevent a disease after a known exposure. So this would be like the thing with Ebola, or people also use it for like HBV virus after a needle stick. This particularly in the case of Ebola, you can use it to lessen the symptoms of an ongoing disease. So healthcare workers that are exposed to something. Another idea with this would also be like a tetanus shot. You can give a tetanus shot, which basically when you get a patient that you think has had a tetanus exposure, you not only give them the tetanus toxoid vaccine, but you can also give them convalescent serum just at a different location. And that would kind of bind up some of the toxin before it had a chance to enter your cells, because it's going to take a while for you to make these antibodies to the toxoid, which is the other part of the tetanus protocol. You can also give it as kind of a prophylactic. So basically to prevent immunodeficient individuals that you fear are at risk for a disease, if there's kind of been a high incidence. So those are kind of the main ways that we would actually use passive immunity. Active immunity is vaccination, and we're going to talk about all of the forms of vaccination in other videos, but just to kind of give a quick overview. You can be vaccinated with a live or an inactive virus. So live would be live attenuated. Basically, I've cultured the virus enough ways, enough times that it's no longer as pathogenic as it once was. So it still replicates, and that's actually really key for a live attenuated virus, but it's no longer going to cause as strong a disease as it once would have. You can have killed whole or subunit vaccines. So like a subunit vaccine, a good example of that would be like the polysaccharides found on Streptococcus pneumoniae. Those have been broken down into subunit vaccines. It's still an antibody-mediated response, with the exception of one, and that's the Bacillus calmette-guerin vaccine. That's for TB. And we don't really use it in the United States, but it does induce a very strong T-cell response, which is actually key to fighting TB. So that's kind of why that one has to be that way. The other videos in this series go into the types of vaccines and then ways of increasing immunogenicity to vaccines.